Another important devices which are frequently used to analyze or to diagnose certain biological conditions are known as the lateral flow devices. Lateral flow devices. And lateral flow devices, LFDs, they are used as rapid diagnostic platforms, and they allow almost instant result from the fluid samples. That is the lateral flow devices which are there, and we can use them as a diagnostic tool, as a diagnostic aid, and very rapid, very precise, and provide the immediate reaction. Now, what is a lateral flow device? This is a form of a strip, can be a form of strip, and for example, there is a port for the sample application on the top of the strip. Then there are the reagents which are required for that in the next. And then through a capillary form, it passes through the capture zone at the bottom, which is created with antibody, which will tend to bound with the complex. It may be a bead complex or others. And then sample moves down from the capillary at the bottom, and it produces a color depending upon the intensity of the diagnosis. For example, lateral flow device, these are simple to use, as we can see, and contain all of the required components within the strip itself. That is the antibody beads, and other material, color indicators, which will give a result. Now, this technology is based on a solid phase consisting of a nitrocellulose or polycarbonate membrane. So you see the strip is, as a matter of fact, the nitrocellulose or a polycarbonate membrane, and they provide the solid phase of the material. A detection zone that is actually coated with a trapping antibody. As we have seen earlier, we have to keep in mind the earlier diagram, that is the detection zone where there is a coated with the trapping antibody and the solid phase on the nitrocellulose membrane. Now, what is the detection antibody? It is conjugated to a solid colored marker. We have to have some indicator, and that is a colored marker, which is usually a latex or colloidal gold, and is stored in a fibre pad which acts as a reservoir. So in the fibre pad which is there, this detection antibody is stored in a small quantity. The sample is applied to the reservoir pad along the sample port. So it is applied to the reservoir pad, which is on the top of the material. Then it goes to the sample port. Now, sample, it can react with the conjugated antibody, which is there, and then the specific antigen, which is prepared or which is present. Now, the sample, it will react with the conjugated antibody only if the specific antigen is present. If the specific antigen is present in our sample, then it will react with the conjugated antibody, otherwise not. And that is the major principle which is there. The liquid then leaves the reservoir, travels up the solid phase pad to the location of the trapping antibody which is present in the reservoir. And the presence of specific antigen which we are testing for. So if that specific antigen is present, then it will react to both the conjugated and trapping antibody. With the both conjugated and the trapping antibodies, it will react. And if the sample is positive, then this results in a colored line. If the sample is negative, then no colored line, it develops there. So it means the sample, the result is very straightforward. It will give us a significant indication, significant diagnostic indication for that. So it means if there is a positive antigen present, then the sample gives the colored line. If it is negative, then no colored line will develop. Now the system lends itself to multiplexing, and up to 3 different antigens can be tested for simultaneously. So you see that it is not only for one antigen. We can also multiplex 3 different types of antigens, and that can be used to analyze different tests simultaneously, 3 types of different tests simultaneously, importing different colors or different material. Now, where this technique can be used? This technology has been applied to many important tests, just like we know home pregnancy testing and various other self-diagnostic kits which are present or which can be used all over there. Now, another use of this technique is used in the law regulating authorities, just as police forces and army, to use for the rapid identification of use of recreational drugs. That is the drugs which are used by the people, which are used by the person. And that can also be used for the testing of those drugs, whether a person has taken that drug or not. And they are, mind you, they are very precise, they are very quick, and very, very rapid methods which are provided for diagnostic.